years of much and we're analyzing.
Good morning, everybody. Thanks for coming, those of us who are here in person and those who were able to join online. It is my great pleasure to introduce today's seminar speaker, Dave Conti. Dave is a professor of Population and Public Health Sciences at the Keck School of Medicine of USC. He's also the Associate Director of Data Science Integration Sciences. Dave has had a long-standing interest in genetic epidemiology and statistical genetics. We met in 2001 in Garmisch-Partenkirchen, where Dave was giving an award-winning presentation on hierarchical Bayes models for pathway analysis, or functional integration.
So
everything else.
And they haven't really moved much further. It's all the same, right?
So Dave has particular interest in genetic epidemiology and modeling in diverse populations, has worked a lot with the multi-ethnic cohort, and is currently co-PI on a project developing polygenic risk scores for cancer that are transportable across multiple diverse populations. And so that's going to inform some of what you're talking about today. So thank you.
All right. Thank you. And thank you all for being here and online. This is fairly informal. So in the room, if you want to stop and ask questions, feel free. Online, feel free to jump in. I can't see the chat, so at least I don't think I can. But feel free to. Oh, you got okay. We're covered. Okay, so I'm going to talk about fairly conceptual type of talk here and the issues of risk prediction using polygenic risk scores, and specifically applying this to across populations and how we might do that. So just generally, what is our goal? Our goal here is to have risk prediction for all individuals, right? And certainly we need to consider the genetic side of things when we come up with polygenic risk scores models and how we would include that and the variation of genetics across populations. But we also have to do this in the context of other factors, right? Cultural factors, people, nations, language, all these other things that go on. And how do then we combine these things sort of in a cogent model? The other thing is just about risk prediction in general. Certainly there are models out there that can be black boxes where you just do the prediction, don't worry about what's in them. But often we want to understand what goes into these models, what do they mean, what do the variables mean, and things like that. And so I'll talk mostly about how to build these models and think about them, but less from a statistical point of view and more just conceptually. So if we think about just the process of developing a PRS for screening, ultimately we want to do this in public health screening in this type of sort of setting. We begin with discovery, right? GWAS summary statistics. Where do we pull those from? What are the populations that they have? Often these are sort of populations of legacy cohorts and other things. What do we use for reference data if we're doing summary statistics? So some of this will overlap with yesterday's workshop for those who attended. So there'll be a lot of similarities with that in terms of just what are the populations? How do we use the methods? What are the approaches? Do we do pruning and thresholding? There's fine mapping. There's genome-wide approaches. There's a plethora of approaches right now. There's many coming out. But it really comes down to maybe the goal. Are we interested in developing a polygenic risk score that is specific to a population? Or is it ancestry agnostic and can work on any individual that way they just come through the door? And so throughout this process, we need to think about what and how we define and use populations through this. Right. The next step is then to take this model, the polygenic risk score, and evaluate it really at the population level. This could be R squared, area under the curve. We often do sort of categorizing and doing quantiles of the distribution and seeing individuals above 10%. What's their what's the increase in risk for those individuals? Same sort of issues though, in what populations and how we do that. We can go to individual evaluation, very similar. We're looking at percentiles of a certain distribution that individual might be in there in terms of a relative risk, but also if we can translate that then to an absolute risk. And what is that absolute risk for the age-specific absolute risk for that individual going forward? And again, evaluation in what population and how we would do that for admixed individuals. And then finally, screening trials. what populations, again, what's the current standard of care? What's a cutoff? What is that modified screening protocol where you say, here's the PRS, here's the absolute risk at a certain age. Do we modify it for this population or that population? How do we do that? So to get started, make sure we're all on the same page. And just to be clear about things, I'm going to go through a little bit of a DAG discussion here on the concepts. Hopefully everybody's familiar with directed acyclic graphs, but this is sort of the world viewed here in terms of what might influence an outcome. And it's very much a geneticist point of view in terms of there's genetic factors that can be both measured and unmeasured here. And then there's other risk factors that can be both measured and unmeasured, right? And we have genetic models that really describe this process with additive effects, and then we throw everything into the variance for the environmental things going forward. Now, what are we really doing on the genetic side, right? We don't necessarily measure the true causal variant. What we measure are markers that are near it and are correlated with that variant, right? That's what's represented by this sort of line here. They're correlated through linkage disequilibrium, population evolution. And so that makes us so we can use markers to capture that effect, and it induces an association of those markers on the outcome of interest, and we can test those. So in a GWAS, we can impute these. They may or may not be the causal variant or the functional variant in that case. And so we can just go through all 30 million SNPs and just sort of do that as a genome-wide exercise. But what's really driving this LD? It's really local ancestry. Okay, so haplotypes being passed down from one generation to the next, coming from certain populations. And so that drives, sort of in a single population, a single measure of LD or correlation that we can measure. But across populations, that local ancestry leads to differential LD effects. So that leads to heterogeneity in measures of LD. Okay, and just to be a little bit clear about sort of the estimation of local ancestry and what we're doing, just for those who aren't sort of geneticists here, the bottom line here is what we would estimate in terms of is the actual ancestry. So in this case, this person has along the chromosome, they have an ancestry mosaic, an ancestry component that came from the red population and one from the blue population. And we want to estimate this, okay, in terms of this line, the second line here, the dotted line. What we have in terms of data, again, the markers along the chromosomes in terms of what we measure in terms of do they have a variant or not, okay, simplifying that. And then we have two reference populations, the red and the blue, individuals that come from those populations. So again, what are we doing? We're defining what a population is, what individuals are from that population. We put them in there, we measure them. We go through this, just a Markov chain, just to give you an idea. Do we see the variant? If that variant is more often found in the red population, then they're assigned that local ancestry at that point. And then you go down and you just use the local allele frequency differences to assign local ancestry. Okay, so the concept of local ancestry exists in terms of evolution and being passed down from generation to generation. We can estimate that through this. So you can get this in terms of 3 individuals here on the HapMax. These are individuals of Amerindian ancestry. And what we're estimating on the y-axis here is the percent of European ancestry at each site along the genome. Okay, and you can see that there's quite a bit of diversity within a population of individuals defined as Amerindian ancestry. Okay, in this case, this is the Hispanic individuals from HapMap. And you can see that this individual has about 50% European, or 1 chromosome from the European ancestry group, and then 2 for the rest of that. And there's different amounts of variations for different people here, right? Now, if we average across all the chromosomes, that can give us a global sort of ancestry component. That's this GS up here, genetic similarity. Okay, and that's related to local ancestry. Local ancestry, though, is just defined by a few different populations in terms of the reference. So it's easier to estimate global similarity or genetic similarity from a set of ancestry informative markers across the genome. So here we're using just allele frequency differences across the genome to estimate ancestry. What do we get from this? We can do this with principal components, and we can get figures like this. Here, just showing several groups from the HapMap populations where we have component 1 ancestry, component 2 ancestry, component 3 ancestry. And these are individuals that, again, are in self-identified groups of populations here. And we can see how they're separated out by these different components in 3 dimensions here. And I'm just pointing out that you can also separate out within European ancestry in terms of Northern Western Europeans versus Southern Europeans. Okay, if we expand this out a little bit, it's really a pyramid. It's sort of the levels of sort of the most common PCs that we estimate in genetic data. And you can see that we have sort of individuals of European ancestry here, Asian ancestry up here in the purple, in the red, it's African ancestry, and over here, Amerindian ancestry. And so we estimate this in terms of a pyramid, and the black dots in this particular case are individuals, or Latino individuals, along that axis between European and Amerindian ancestry. It's an admixed population, or individuals that are admixed. Okay, so we have this variable to think about as we go forward here. Okay, but that variable itself is related to race and ethnicity. As we saw in that picture, just talking about ancestry using genetic similarity, it's related to race and ethnicity and self-identified. So there's a correlation there. And then, of course, we know that race and ethnicity are related to our non-genetic factors. So in creating this whole figure of the DAG, with that relationship of genetic similarity being related to non-genetic factors, okay, that induces a correlation of genetic similarity to those non-genetic factors, but also to the outcome of interest, right? And that's why when we typically do genetic studies, we adjust by genetic similarity, control for potential confounding by population stratification. So this is the simplified confounding triangle and why we adjust all the time by genetic similarity, control for confounding, okay? So what we realized early on, I think, in GWAS is that there was potential confounding, and there's this differential LD component, and that drove, I think, the field to think, okay, well, we need to limit the differential heterogeneity in this variation and really just stratify by race and ethnicity. And so the outcome of that was, due to a lot of other factors, really led us to a lack of diversity in current GWAS studies. And this is a paper that I think gets shown a lot in any of these conversations. It's a great paper. It just shows over time here on the x-axis the growth of GWAS studies and the number of individuals in the GWAS studies, but you can see this red component here far outweighs that in terms of the number of individuals, and those are individuals of European ancestry. And they definitely overrepresent the global population distribution. What's the impact of that? As we discover variants and we construct polygenic risk scores from that, that will impact the transportability of those to other populations. So what this shows, we have populations along the x-axis and the predicted accuracy relative to Europeans on the y, and you can see in all other populations taking a polygenic risk score that's developed in just any GWAS today, it will perform less well in other populations. Okay, and particularly poorly in individuals of African ancestry. Why is that important for a lot of what I do? I do work in prostate cancer, and prostate cancer has, in terms of incidence and mortality, about a 1.5 to 2-fold increase for individuals of African ancestry and African Americans, okay, relative to white individuals. The other thing to note here is that Asian individuals have about half the incidence for prostate cancer. So in this context, what we did out, and this is really a lot of work that's led by Chris Hyman at USC's, compiling sort of all the world's data on prostate cancer and really focusing on diverse data sets and having different ancestry. And so just in terms of what we've done in the last year, this paper was just recently published, is sort of increasing the diversity and increasing the sample size in GWAS studies. So we have, again, we do it in terms of 4 different continental ancestries. Why? Mainly due to history in terms of how consortiums and populations and things were constructed in terms of gathering the summary statistics. This is the data that we have today, okay? And we can't often go back and get the individual level data from all these studies. So we have to deal with sort of bins of continental ancestry at this moment. So we have European, African ancestry, Asian ancestry, and then Hispanic ancestry. In this particular example, we have 11 new studies, which is a 57% increase from the previous GWAS that we performed in terms of non-European cases, and then over 128% increase in the sample size overall. Now, just to be clear about what do we mean by a genetic risk score, a polygenic risk score, what we're really doing is just summing up the number of risk alleles, okay, represented here by G. We don't treat all of them the same. Some are more important than others, so we weight them, right? So it's just a linear combination of weighted risk scores. And the question really of interest is what SNPs to include, and of course, what weight, right? And then thinking about that across populations. And if we go back to our DAG, the question with the construction is do we aim to pick all those putative causal SNPs? Is our goal to really just pick those out and say this is the best thing? That might put us on the side of sort of multi-ethnic fine mapping. Or are we just really interested in sort of weighting the entire region? I don't care what SNPs are really driving this. I just want to weight it in an appropriate way. Okay, that then now I need to take into account the linkage disequilibrium and also local ancestry potentially. What then does that mean across populations? Do I have a different set of SNPs? Do I have a different set of weights? Okay, and again, if I develop a different set of SNPs and weights, is that a population-specific PRS? I have to put people into bins for that. Or is it an ancestry agnostic type of PRS? So what we did is we discovered 451 prostate cancer variants. We're really focused on an ancestry agnostic polygenic risk score, where we did multi-ethnic fine mapping from some methods that I talked about yesterday in AMJAM. And I, again, don't want to focus on the methods here too much. What we did with this method, though, is we have a single set of SNPs across all, working for all individuals. And so though we preferentially selected SNPs found across all populations, okay? Although there are a few SNPs here that are population specific. And then we used a multi-ancestry weight. So weight that was from the meta-analysis of all ancestries for that. So we're preferentially weighting towards similar SNPs with similar effects across populations. Okay, here's the distribution of that within the population bins, okay, when we look at that. And you can see that there's a shift in the African Americans of this PRS, if we just look at the raw score as we create this, right? And we see that shift, and we also see that the Japanese Americans, in this particular study that we've done, slightly shifted downwards. Okay, so we're really tempted to interpret this distribution as reflecting that incidence that I showed before. Well, we've picked out the causal variance, we have a nice polygenic risk score distribution, and it's reflecting what we believe to be incidence, okay? But please don't do this. There are many caveats against that, okay? We would like to do that. We need methods that maybe could get us there, but we're not there yet. There are several different studies that have shown that if you look at the distribution, you see sort of differences in populations, and they don't reflect sort of incidence rates. And this is just an example from this paper by Reisberg, where we're looking at Ph.D. and type 2 diabetes. Actually, the patterns of the distribution of these polygenic risk scores are very similar in terms of the polygenic risk score, where you see that the one with the upper level here on the far right is from African American individuals, or African ancestry individuals, and the one on the left there is common is sort of individuals of European ancestry. So, and it's due to linkage disequilibrium, and they specifically say as the linkage disequilibrium between the causal and associated SNP varies in different populations, the effect size of the disease-associated variant, that test between the marker and the outcome, will be overestimated in non-Europeans. That's what we're using as the weight. Right? But we think, okay, well, we're using a multi-ethnic weight, maybe we're getting around this because the only thing then that's driving population differences are allele frequency differences, not the weight, right? Well, SNPs in our models tend to have higher effect allele frequencies in African populations, and that could be due to evolutionary aspects and other things going on, okay? And again, it depends on how we constructed this polygenic risk score. Are we weighting markers, or are we trying to aim for the causal variant? So typically we just standardize within populations. We define a population, we standardize, and we get standard normal and everything's but standardized so we can go forward. We can then look at the performance of a polygenic risk score by looking here on the x-axis, the odds ratio per one standard deviation increase in the genetic risk score. Now, what I'm showing is sort of the evolution of our polygenic risk scores across time, given GWAS that we've done over time. So around 2014, published a paper that was multi-ancestry, but predominantly 95% European ancestry. We ended up with 100 SNPs in that case. And you can see the performance of that. Here's the green dot on the right-hand side across different populations, European, African, Asian, and Hispanic. And then the most recent one here, the 451, where we've really focused on making this multi-ancestry and including the largest non-European populations, at least for cancer. And you can see that there's an improvement across time. Sample size matters, diversity matters, okay, in terms of where that is. And so in many regards, what we're seeing in other sort of traits and cancers where a polygenic risk score does not translate across populations, at least in prostate cancer, it does perform fairly well in all populations. Yes?
Sorry, but when you do bin people into different racial groups, however you do this based on ancestry markers or self-report or whatever, did you see a huge difference in the weights for these SNPs? That's
a great question. I have showed this figure yesterday in the workshop, so I could. We see about around a 50% correlation between them compared to Europeans. The problem is our European sample size is much larger than these other studies. So is a lack of sort of correlation in these estimates due to a true effect being different across populations, or is it due to just stochastic noise? Because we have subpopulations have much smaller sample sizes, things vary a lot in that context. Do we truly believe that there are common effects, alleles across populations that have differential effects? Probably not in terms of a main effect. Is there an interactions? To date, there's very little evidence for replicable interactions for SNPs, right? But that's one of the assumptions I get is in going in with this. Okay.
Follow-up question.
Yeah.
How do you define the weight?
Oh, so in this case, they're just the log odds ratios from a meta-analysis across the population. So we have 4 summary statistics from each population, and we do a fixed effects meta-analysis to get that weight. So it's a log odds ratio. Although I'll come back to that here in later on, modifying that a little bit. So how can we use this for absolute risk? Okay, and this is just a simplified conceptual thing of absolute risk, right? We have the risk in the exposed. It's going to be equal to the risk in the baseline group, okay, multiplied by the relative risk. Okay, we have a group that we're saying is the baseline, and from that, we're going up in terms of what is that relative risk, right? To get this in terms of using reference populations, okay, we can do this and define the baseline risk in terms of ethnic-specific incidence rates and ethnic-specific mortality rates from SEER. Okay, and we can pull down that data for prostate cancer, and then we can use the relative risk from the PRS estimates that I just showed. We can use ancestry-specific, or we could use the multi-ancestry type of averaged over all of those, which is about 2-fold increase per 1 standard deviation. Okay, and we can create figures like this in terms of cumulative absolute risk, age-specific. Again, on the age here, on the x-axis, absolute risk over time. And if we look at the distributions here, these are the standardized distributions between Africans and Europeans. And what we're going to do is define the cut point for those populations where all individuals above that cut point have a 2-fold increased risk or greater. Okay, so that defines what these lines are in terms of their overall relative risk, the 2-fold increase in risk. Then we put that in terms of the baseline risk, and we can draw these figures for those individuals. And we can see that for European individuals, the lifetime risk is about 7.8% from this type of analysis. And individuals that have 20 at the 20 percentiles of the PRS here reach that threshold 16 years earlier. Okay? Individuals of African ancestry reach the lifetime risk, which is higher in African American, okay, at 19 years earlier, about 66, age 66. So that has implications for when to start screening. If we knew what clinically relevant sort of cutoff for absolute risk is in prostate cancer. Okay, but the question here is how to do this for admixed individuals. Now, certainly what we can do is we can standardize the distribution by a theoretical population that's similar in terms of the admixture. That's something we can do. There's ways to do that. What we can't do with reference data, at least from SEER, is for an admixed individual, what's the baseline risk? Okay, where there we need individual level data. So Good. Ruth, thank you for joining. I've referenced your
Oh,
book
thank you.
here. How do we do this for individual level data, right? We estimate absolute risk with individual cohort data. Most common is cause-specific hazards, Cox model being one of them. But I just want to focus that this is the same sort of thing in terms of we identify the risk by a given set of covariates or risk factors compared to a baseline with that set, okay? And then you have the relative risk component. That's just one model. There's a lot of linear, and again, we don't need to focus on those. I'm just sort of outlining this as a way forward. Now, the data that we have available to do this is using our multi-ethnic cohort. So this is a cohort that was started in 1993. It's 200,000 individuals across 5 different ethnic groups. Over time, we were able to sample in about 2001, 34% of them to enter sort of the MEC biobank with a blood sample. Of those, we were able to finally GWAS them now, 20 years later, or all of them, through a set of nested case controls and other types of studies. And so we have a genetic database of about 73,000. This just gives the number in terms of what we have available for colorectal cancer, breast cancer, and prostate cancer. If we remove prevalent cases starting at the time point of the biobank, going forward, we remove some of those individuals there, and this is the sample size that we have available for prostate cancer, about 2,800 cases, incident cases from that time point on. Just to give you an idea, they're sampled from Los Angeles and Hawaii. Most of the African Americans and Latino individuals come from Los Angeles, and Native Hawaiians, Japanese Americans, and white individuals come from Hawaii. So in going forward and thinking about how we can model this, we really need to be aware of sort of this recent report by the National Academies on using population descriptors in genetics. Okay, and it's a good report. I suggest everybody read it. It's confusing at times, and I think the field is really thinking about how we use their recommendations or the ideas of the recommendations actually in practice. And so just highlighting a few things here. Recommendation 1, researchers should not use race as a proxy for human genetic variation. We could also say researchers should not use race as a proxy for non-genetic variation as well, right? That goes into the fourth recommendation here. Researchers conducting human genetic studies should directly evaluate the environmental factors, the non-genetic factors. We should aim to do that. But then when we go back to sort of how we use these, we need to tailor our use of these population descriptors. And they give sort of a general table there, sort of describing different types of genomic studies. What should we be using in that context? And that's really what I want to focus on here is what we're doing in this context. I say we might fit into category 4 and 6 here: trait prediction, complex traits, and then health disparities with genomic data. Here, the plus is under this genetic similarity column in using genetic similarity for genetic research when we use that, right? And then I'm going to talk about, well, what do we really do when we use that in these types of models here? And so just a little bit more background on the MAC and the population descriptors. And I realize this is small to see in this, but just for those in the room, self-identification, right? So we have a questionnaire. They can identify one or more of these. Most individuals in the MEC in 1993, when they started, were 40 plus years old. So current day, they're actually, they're an aging cohort. So we have Black and African American, Chinese, Filipino, Hawaiian, Japanese, includes Okinawan, Korean, Mexican, and other Hispanic, white or Caucasian, and other. That's the data we have from the questionnaire. Okay. We can refine this a little bit in terms of the race variable to capture multi-ethnic backgrounds, or if they measured or if they selected one or more bubbles on there. So all races, if they responded, and we ordered them in terms of those categories: Black, Hawaiian, Latino, and so forth. So for an example, if a person selected Chinese, Hawaiian, and Samoan, they would have a new variable, this sort of more detailed variable of being Hawaiian, Chinese, and other, okay, just to be clear. Now, of this, individuals of this generation, in general, I think, are hesitant to identify as multiple categories. I think that would be different in today's world, so we'll see that as we go forward. And one of the things that we also do is to estimate this genetic similarity. That's going to be dependent upon the populations we use to estimate that genetic similarity. So in some sense, for full disclosure, here's what we use to estimate that. It includes the 1,000 genomes, but it also includes more diverse sort of populations, individuals from populations from the PAGE Global Consortium, the Global Reference Panel. And then we also categorize those in terms of superpopulations, continental ancestry, but also subpopulations, getting into a little bit more nuanced in terms of West Africa, South Africa, also sort of different groups within East Asia, okay, as we go forward, just for reference. So one way to estimate genetic similarity, and often called, is through clustering. Just to be clear, statistically, what we're doing, we are picking the number of clusters, and then we are estimating how these individuals fit within that clusters. What we've done in this figure is then order them by self-identified race and ethnicity, and we align on the y-axis there is the probability that that individual fits into a cluster. And in this case, we have clusters that cluster 1, cluster through cluster 5. Now, when we look at a picture like this, we see that the cluster that we've colored red tends to be more often in individuals of African ancestry, and then we label that African ancestry. Okay, I'm going to avoid labeling it that and just call it cluster 1. Okay, we can do the same thing in terms of assigning ancestry within the different clusters or different race and ethnicity groups, but we can just leave it as cluster 1. Okay, and again, this all goes into sort of should we label these or not in terms of just is it ancestry or is it just similarity that we're estimating within groups of individuals that we've a priori defined within populations. So another way to do it is through the principal components. Again, we don't label things, we just look at components and then we sort of plot them and then color the individuals in terms of their population labels here. So what I've shown in this figure, these are the 5 different plots for the 5 different groups of the multi-ethnic cohort. All what's similar across all of them are the sort of anchor populations or the reference populations here. The green is the European, red is African ancestry, we have Asian ancestry in purple, and blue is Amerindian ancestry. And so we can see here that there's, for individuals who self-identify as white, there's quite a bit of diversity on the different axes as well. I do want to point out this sort of group that's in the middle here that's about 50% equidistant from European ancestry and Asian ancestry, first generation biracial individuals that self-identify as white. Compare that down to the below, the Japanese Americans. There are a lot of individuals there within the sort of Asian ancestry, but we also have a group of individuals that are about 50-50. So genetically, they're similar, self-identified differently. Think about also the time period, right? For these individuals at 1990, about 40 years old, post-World War II, are they willing to self-identify as non-white or what, right? So again, thinking about sort of the social context of how individuals self-identify, we can clump them together genetically in terms of similarity, but does it mean the same thing? Just pointing out some other things in terms of African Americans, we see diversity from African ancestry to European. Likewise with Latinos from European to Amerindian ancestry, and then Native Hawaiian at this level from European to Asian ancestry. We can also look at this in terms of other factors a little bit more detailed within one of those and sort of higher level principal components. Again, I'll point out that just, the majority of individuals self-identify as one category this time, even though we do see that there are a lot of diversity within sort of this group of white here. And we've just separated out the colors here in terms of the MEC. The sort of aqua green color is white individuals sampled from Los Angeles. The gray here is MEC individuals white sampled in Hawaii. And you can see those that self-identify as white sampled from Los Angeles are more likely to have that Amerindian ancestry, okay, or admixed with that Amerindian ancestry. So again, what happens when we use genetic similarity to capture that risk versus self-identified race and ethnicity? Also, if we go forward in terms of African Americans, we can see that most of that genetic ancestry really aligns with West African ancestry, given the population, age, and time things. You can see that in differences across the US. We can see different sort of ancestry within Africa when we look at individuals that self-identify as African American. If we look at Japanese Americans, just showing some of the diversity within and the admixture that we have in terms of Japanese white, Japanese Chinese, Japanese Filipino. This axis along here really captures sort of mainland Japanese versus Okinawan Japanese, and individuals that we could identify through Okinawan surnames and other ways of doing this. And then, within our Latinos, we do see quite a bit of diversity there, but most of it is from European to more Mexican Amerindian ancestry. Okay, so most of them are sort of from Mexico, or at least heavily influenced by Mexican ancestry. And then finally, Native Hawaiians, quite a bit of diversity. And this is just in sort of higher levels. And this is Asian ancestry and European ancestry. What is most likely a Polynesian type of ancestry component there on the top part, but you can see that there's quite a bit of a diversity for all individuals within this group. Okay, so now that we're sort of understanding the genetic similarity related to race and ethnicity categories, how do we model absolute risk? And the first thing we can do is to stratify our absolute risk models by race and ethnicity. So just look within each race and ethnicity group, do a simple sort of model of the polygenic risk score. This is the effect, okay, of the polygenic risk score in the MEC individuals across all populations. It is close to twofold increase, although we do see sort of a lower estimate within African American. So not quite what we were hoping in terms of sort of being common across all populations, but not bad. So the differences in race and ethnicity are captured a lot by the sort of baseline risk. And if we look at sort of cumulative risk plots across different race and ethnicities for the 20th, 50th, and 80th percentile of the genetic risk score, this is the pattern we have. And you can see that increase in African Americans relative to all other populations. So that's defined by the baseline shift. And then the split between the three lines is defined by that relative risk, okay, that's driving that. Now, the advantage of this type of data is that we don't have to put people into bin categories, so we can actually just put them all in one analysis, estimate a common PRS effect, and then look at how how race and ethnicity would change as a relative risk, not the baseline. So our overall PRS effect is 1.92 across all populations. And now we have an effect estimate that compares sort of race and ethnicity to the reference group white here. So again, that baseline, now the baseline is common across, but we have a relative risk that captures race and ethnicity differences. That's showing this difference.
Sorry, but the relative risk refers to a continuous PRS increase, or is this in data?
It's continuous PRS.
Okay.
Yep, we're modeling as continuous PRS. Yep. And then we just give cutoffs of that right here. So again, what about admixed individuals? We could now model genetic similarity in here as a, sorry, I can't see it on my screen here, but as a relative risk factor, put that in as the model. Okay, we have a common PRS and then a genetic similarity factor in the model, so common baseline. We can put that across here. And this is a little bit complicated, so let's focus here on the African Americans. What I'm plotting are the red and green lines come from the model that I just described. Put in genetic similarity, and then at the level of genetic similarity where there's 100% cluster, those are the red lines. In terms of the ancestry now being a continuous variable, so we can put that at that, and then the polygenic risk score being a continuous variable, so we can put that at percentiles. These are the lines we get. We can choose different values for that, and the average across African Americans in terms of admixture is 85% cluster 1 and 15% cluster 4, right? So that's the green line there. That's being compared to the model that I showed previously where we modeled these things just by race and ethnicity, and that's the blue line here. So you can see that modeling by ancestry is different than modeling by race and ethnicity, and now we have the ability to do this by a sort of continuum of ancestry. And we can do that across all sort of genetic similarity ancestry groups, right, and interpret them in this way. But what are we capturing with this, right? What is this genetic similarity? It's an admixture of an individual defined through allele frequency differences. But what we're really capturing are distributions of non-genetic risk factors, not genetic factors. Okay, so we're using a genetic variable, genetic similarity, to capture correlation of these non-genetic factors. So what we really should be doing is trying to aim to capture these factors, right? Really put these factors in the model. And so we look at joint models and looking at the distribution of these known factors to capture that risk across populations. And here I'm going to go back to a paper by Malcolm Pike in 2004, who used the multi-ethnic cohort to do this analysis. Same sort of thing, looking at breast cancer across populations. He used a log linear model, and in this particular one, common baseline, using a relative risk that's a function of race and ethnicity. And we see that relative to whites, African Americans have lower risk, Native Hawaiians have slightly elevated risk. Okay, so this is the way to look at sort of differences in a single population driven by that. Now we can look at the impact of adjusting by known factors. And here's the list of known factors. So we're going to put in the race and ethnicity variable and now adjust for known risk factors in breast cancer. What do we see here? We see that when we adjust for the known risk factors, the effect in African Americans by the variable race and ethnicity goes away. There's very little difference between those two. For Native Hawaiians, it actually increases. Okay, so the conclusion here is the risk factors adjust out the effect of race and ethnicity for African Americans, but it doesn't do that for for Native Hawaiians. Another way to look at this is, can we use the distribution of the risk factors to predict what the risk should be based on these risk factors, and then compare those across populations? So what we're doing is we're saying here, we know what the relative risk is in the entire cohort for these risk factors. Now I'm going to use the distribution in every population to give me a risk prediction. What is the risk in that population? And then I'm going to compare the predicted risk in white individuals to the other populations. Okay. Nowadays, this is very similar to causal modeling and almost G-estimation type of approaches. Doing this, we get sort of comparing just the predicted risk based on the distribution of the risk factors. We get African Americans have lower risk, Native Hawaiians have lower risk. So that's very different when we compare to sort of the observed risk differences within those populations. So again, the conclusion is the risk distributions in African Americans explains that, but it doesn't explain the Native Hawaiians. So interestingly, Malcolm went on in the discussion section of his paper to say, the most interesting finding is the very high breast cancer rates in Native Hawaiians, the distribution of the number of high-risk alleles, and this is based on 2 loci at that time, okay, is different in women from different racial ethnic groups. 42% of the Native Hawaiians have 3 or 4 high-risk alleles, where only 25% of women in the other risk have those high-risk alleles, right? This could be a significant contributor. But what is he doing? He's using a polygenic risk score, the distribution of a polygenic risk score, suggesting that that's driving the differences, right? And this polygenic risk score here is 2 loci, but he's using it to say that it describes that difference. So can we do that for prostate cancer? Okay, ignoring my advice from earlier and saying, let's try that. Okay, and just sort of what we're going to do is modify this a little bit and use a different kind of weight. And what this weight allows us to do is it's just a weighted sum of the alleles in this case. And so now the distribution, it's not weighted by the log odds, but the distribution can be interpreted as the count of risk alleles in the population. Okay, in terms of if you have 1, 2, 1, 0, 1, or 2 for every individual, this is sort of the weighted average of the counts of those distributions in the population. And we can see that we get the similar sort of distribution differences between populations. But now when we model this polygenic risk score, the corresponding estimate corresponds to a 1 allele increase. Okay, so if we model that combined in the population here, we get an increase of odds ratio or hazard ratio, of 1.01 per 1 unit increase of a risk allele, right? So that's not very informative. What we can do is look at the population differences here between sort of African Americans and our white individuals. It's about 12.5, so we can modify our risk effect that way. And we see that If we use the distribution of the polygenic risk score to describe differences across populations, that difference is 1.13 between white individuals and African Americans. Visually, that looks like this. So the distribution of our polygenic risk score does not explain the differences that we see in populations, right? And this model has very poor corresponding fit statistics if you look at it, okay? So where does that leave us in trying to model these things going forward? And just as an example, and going back to breast cancer, the distribution does not follow incidence at all, okay? We see when we look at it in the MEC population, white individuals have a lower, okay, polygenic risk score than all other populations. And oops, just to highlight, we will go back to our estimates that we had from the Malcolm Pike's paper, African Americans actually had lower risk. So we can't use this polygenic risk score distribution to define incidence in breast cancer. So overall, where are we and what can we do? The overall conclusions are, hopefully I'm demonstrating the importance of a diversity within a single prospective cohort. We can ask these questions only because we have the multi-ethnic cohort. It's a diverse cohort that's sampled in the same way, and we have genetics on that, right? Often when we make comparisons across populations, we have a cohort that's defined by one race and ethnicity, and we compare it to another cohort. And then there's all sorts of different questions of generalizability and comparability when we do that. Then there's this question about the potential for joint modeling. We need to be careful when we use population descriptors. Is it okay to use genetic similarity? How do we interpret it? In another study that we're doing where we looked at genetic similarity on mortality in the MEC, this was a certain collaborator, and the first response was, well, this is capturing overall genetic effects. The genetic similarity variable is most likely not capturing any genetic effects. It's capturing non-genetic effects. And is it capturing it in the way we think that we're capturing it, right? And should we be using proxies for that? Other proxies, and that just gets to exposome or social determinants of health and other things. Now, the challenge for prostate cancer is there's very little evidence for non-genetic factors in prostate cancer. There's lots of evidence for genetics, not a lot for non-genetic factors, but should we be looking at social determinants of health? The other problem with using social determinants of health, they're often defined within populations, and they're really sort of not interpretable across populations. Think of redlining certain racial or ethnic groups. It doesn't happen in other racial or ethnic groups. How do you then use that variable to describe differences across populations, if you can? The other thing to think about is the additional considerations for residual confounding or interactions when using any of these variables or a polygenic risk score. Just to highlight, for the polygenic risk score with prostate cancer, and this is common across many other cancers and traits, we see an age interaction. So if we just stratify by the age of diagnosis for prostate cancer, greater than 55, younger than 55, we see that the effect of the genetic risk score is much larger in younger age of onset individuals. So that's something that we also need to consider sort of when we construct these models. And age is just one example going forward. So the last thing here is when and how we can use a PRS distribution, if we can at all, to capture risk differences. It depends on how we construct that, right? There's a lot, sort of the best methods for risk prediction right now in terms of performance accuracy are these genome-wide approaches that just sort of weight across the genome, weight across the LD, and you don't really interpret the variables. So we're not capturing the causal variant. So the distribution then is even sort of less interpretable in that sense. But if we focus on getting just causal variants, maybe we can interpret it. So something to think about. So I will end there for questions and just thank, a lot of the prostate cancer work here. I'll have to thank Chris Hyman. David Bogomil is the one working mostly on the absolute risk models right now, currently. So thank you very much.
Thank you. And that's actually a reminder. I wanted to also thank Dave and some of his team for the presentation yesterday in the workshop. So David and Sylvia did a great job developing some hands-on code, which is available on our on our course website for people to check out if you're interested in developing polygenic risk models that work well across multiple populations, or doing fine mapping, multi-ethnic fine mapping. So all that's there. So we'll take one question in the room and then we'll go to the chat.
Okay, excellent talk. Thank you very much. I want more, a little bit broader question. So what's your thought on using SEER race-specific incidence rates then? Should we throw those out, or are they still useful?
No, they're still useful, but we have to go through the process of looking at data and calibrating, validate, and looking at these models to see. They give us a indicator of what are the overall levels, right? If we look at African Americans from SEER data and compare that to the MEC, SEER rates are higher in terms of the baseline. Okay, so our MEC African Americans are different, right? So this is always an issue in comparing cohorts, is the generalizability, and we just have to think about that in terms of SEER rates as well. But we have to be just aware of the binning and where these individuals come from, right? That's my simple answer for you. But we can talk more about, yeah.
Okay, and if folks online want to ask the question live, just raise your virtual hand and we'll call on you. But in the meantime, How You has a bunch of questions. The first is, if we consider a PRS composed of an independent set of SNPs,
An infinite set of SNPs.
Independent.
Oh, independent set of SNPs, okay.
And you pick one allele at each locus, right? And you assume that the effects are consistent across ancestry. Is it valid to interpret observed differences in mean PRS values as differences in incidence rates that are attributable to genetics?
It's a good question. Maybe for some diseases we can, if we can do that. It's sort of, I think what you're getting at is, if we could identify the true causal model, right? The true set of causal SNPs incomplete, right? We have the oracle property and we can do that. Then yes, you can, I think. We're not able to do that. At some point, you're grabbing noise, right? If you do this across the genome enough. And as you start to grab noise, you're starting to grab ancestry, I think. And so then that distribution becomes more of an evolutionary process and less of a risk process, even though you might identify them, right? So in an infinite sample size, and you could really, the oracle property, then I think we lean towards it, right? That's where I'm sort of hedging of like, so one of the things we're thinking about is, well, what if we use the top 20 SNPs in prostate cancer that we just truly believe for, many reasons, that these are the causal ones? What do we see then? What does that look like, right? Because when Malcolm made his statement about that, there were two good belief about those two. Well, did that show things? Was that interesting, right? So that's my feeling.
And full disclosure, I had already popped in a response in the chat, which is very consistent with what you said.
Okay. You set me up.
Yeah, but there's Alicia Martin's paper from 2017 goes through exactly these discussions.
Yeah.
And empirically, so there's like theoretical reasons for why we might be like, the true effects might be the same, but our estimates are noisy, and that's going to lead to funky things.
Yeah, even in the case of using true effect, like even estimate, yeah.
So in prostate cancer is kind of the exception, where the mean differences in PRS line up with sort of known differences in risk across populations. Yeah. Breast cancer, lots of other traits, things are like the mean differences in PRS sort of have nothing to do with.
Yeah, most don't even line up, and so it's not even an issue to think about. It's just in prostate cancer, they do, and they have for a while, and it's interesting.
Questions from the room?
Well, why do you think that is?
I think it's because in prostate cancer, the 8q24 region drives a lot of the risk. And in 8q24, that was a region that actually was initially found through admixture mapping. There are big differences in terms of allele differences by African ancestry locally. So any risk allele you pick from that will be different and in higher frequency in African American. So that drives that shift. Interestingly enough, though, if you look at Asian alleles, Asian populations and allele frequencies in that region, they're also higher. So it's the rest of the genome that drives some of that down in terms of the distribution. So it's, but that I don't know. But I think that's why we see in prostate cancer, African Americans distribution driven up.
Even a broken clock is right.
That's the less sophisticated way of saying what I just did because I'm standing here.
Never mind my cynicism. So another question from the chat. This is from Hanyu again. In the context of clinical screening for high-risk groups, which way is more commonly used, using absolute risk measures or relative risk criteria for selection? So I guess identifying high-risk folks, relative risk or absolute risk?
Well, I think the goal is to put it in the context of absolute risk, because that's at least a little bit, I think it's more interpretable for most people. Clinically, there are sort of absolute risk values for certain these breast cancer and others that they have kind of well-established levels. So, I mean, I think that's what's more common, right? To answer that question. You know, again, looking at the age-specific ones, the question, you know, I always come back, and I think I have said this to him when he asked the question yesterday. You know, with breast cancer, we have sort of known variants that have high risk, BRCA1, BRCA2. You know, we know these variants give a lifetime risk of over 80%, right? So a person wants to know whether they have this. Making a decision when you're 20 is very different than making a clinical decision when you're 40 or 50 and after reproductive age, right? What do you do when you do sort of major, you know, surgery, preventive surgery? And so that's really the goal of defining this absolute risk is by age, right? We don't necessarily want that relative risk because that's the same across all time points. We want to know at a particular age, what's my risk? And over the next 5 years, is it great enough that I need to make a decision?
Any other questions?
Everyone, out of curiosity, so these causal risk profiles associated with types of cancer, do they tell us anything about the etiology of that particular type of cancer?
That's a that's a great question and a ongoing. I mean, the short answer is yes, they do tell us some etiology of that, right? I mean, again, that goes back to the goal of defining these causal variants for sets of these credible sets is to give them to, you know, a lab to go in and further understand mechanism and the biology, right? So, so I think that's, you know, part of it. If you look at just the SNPs we pull out and do a pathway analysis, there isn't much evidence for any one biological pathway in this context to say, oh, well, this is really driving it, because it's 451 SNPs. They're not annotated. Some of them are not in genes. How do we do the annotate? There's lots of issues there, right? But, you know, DNA repair keeps popping up in a lot of these things across lots of cancers. So I don't know. I mean, Pete might have an added comment, but, you know.
Okay. Well, let's thank the Epidemiology. Thank you so much. And thanks for your visit. And everyone, have a good day.
So this language is in the little office of competence.
Oh, okay.
But if there is forget lots, just remember that movie is some of those. Good.
Thank you.
Just curious your thoughts on how far we are.
Thanks for letting me crash.
Oh, yeah. Thanks for coming by.
Sure.
Should have.
As we decades.
Well,
Well,
You
you
know,
know,
Yeah, it's an interesting question. People are using it.
Yeah.
We need it.
Oh.
So what is clinic?
How's it going?
So can someone who already